Director of Asset Management.
Director of Asset Management at UniQuest. So it's a joint presentation from Associate Professor Sumaira Hasnain and Dr. Ben Phillis. That's correct, yes. To tell a story of a really interesting translational success that's come out of MARTA research, from basic discovery through to starting to get it into the commercialisation phase. And Sumaira has an NH&MRC CDF fellowship still. She's still on the old CDF fellowship scheme, and she's been a group leader at MARTA research for about 4 years. 2018, and she's been running a really dynamic group at MARTA Research with focus on epithelial cell immunology and the interface of disease and the epithelial layer in the lung and gut. And so this is a story about how they've partnered with UniQuest to actually achieve investment and spin-out company to translate some of her early discovery findings. That's about right?
Yes, right.
Yes.
Thank you, Ali, and thank you for the invitation to speak today. So I was just going to introduce the basic side of things, so basic research first, and then Ben will join and talk about the commercialization side of the research. So I guess the basic science discovery started from my interest in the cellular stress response, and we were interested in what happens to a misfolded protein in different diseases. And what our discovery is based on is the fact that we discovered cytokines can induce this pathway, so they can initiate ER stress in a cell, and this can result in protein misfolding. And this is a part of several diseases. So for instance, in inflammatory bowel disease in the gut, you get protein misfolding because of the cellular stress in the gut, making the mucus barrier thinner so that your microbes can come into contact with your epithelial cell layer and cause inflammation. And so similarly, in a pancreatic beta cell, which is responsible for making insulin, and that's hampered in type 2 diabetes, for instance, and that's what we were really interested in, and how cytokines did that. And what we discovered was that not only did cytokines induce this pathway, there were a couple of cytokines that actually suppressed this pathway. So this just meant for us, opened up an opportunity where we could potentially use these as a therapeutic. So what we discovered, you don't need to look at the detail here, is just the ER stress pathway. And what we discovered was, by doing an unbiased approach, which is shown on a heat map here, that a couple of the cytokines, so IL-10 and IL-22, were the ones that suppressed stress. And so potentially this opened up an opportunity for us to use it in as a therapeutic. And why we decided to focus on IL-22 is because it was highly potent, and also because its receptor is specifically expressed on pancreatic beta cells. So this is what we knew about IL-22 at the time. And if you look at human GTEx data, we saw that the pancreas are the most highest expressors of the IL-22 receptor. So this was really interesting because the pancreas has secretory cells like pancreatic beta cells, which are in charge of making insulin. And this is diminished in type 2 diabetes. So when we looked at IL-22 receptor expression on pancreatic beta cells, we saw that it was really high as well. So we decided to use IL-22 as therapy. And so what we did first off was to use a type 2 diabetic mouse model, or it was a mouse model of pre-diabetes, really, put animals on a high-fat diet and then treat them with a couple of different doses of IL-22. And what we observed was that IL-22 really potently normalized random fed blood glucose in these animals. So this sort of highlighted that potentially IL-22 could be used as a therapeutic in type 2 diabetes. We also looked at immunohistochemistry to look at what was happening to the insulin. So we show that the blood glucose levels are normalized. We wanted to see what was happening to the insulin. And basically, the conclusion from this slide is that IL-22 suppressed the cellular stress response so that your insulin can fold better in your pancreatic beta cells and be secreted. So it's high quality insulin that you produce. And therefore you have lower blood glucose levels and it fixed the hyperglycemia and proinsulinemia that we see in type 2 diabetes models. We also saw some effects of IL-22 on satiety. So animals ate less, and we saw some effects on appetite in general. And importantly, what we saw was that IL-22 administration improved lipid accumulation in the liver as well. So in type 2 diabetics, especially the ones that have progressed a lot, you get fatty liver disease. And in the high-fat diet animals, you can see the lipid accumulation in the liver in red. And this was reduced significantly when we treated the animals with IL-22. And these are just pictures of the liver. You can clearly see that the high-fat diet livers are very fatty, white in color. And then when we treat with IL-22, these return almost back to normal. So this is the problem, and the reason why I showed you why IL-22 has lots of different effects in the mouse, for instance. The IL-22 receptor is really highly expressed in the gut, in the skin. We saw effects in the liver, and we obviously saw beneficial effects in the pancreas as well, but the problem is that it can induce hyperproliferation in the skin and the intestine as well. And our lab's shown that IL-22 increased proliferation of colon cancer cells. There have been studies with human samples where they've shown that in colon cancer, you get really high expression of IL-22 and the IL-22 receptor on epithelial cells. So cells that can divide really fast, like the cells in the intestine and the skin, IL-22 can induce hyperproliferation in those. And this is connected to IL-22's effect as a wound healing cytokine, basically. So when there's an injury, there's an increase in endogenous IL-22, which leads to wound healing, but this just means hyperproliferation. So you can imagine that if we give a chronic dose of IL-22, like you would need to for type 2 diabetic, for instance, this could lead to hyperproliferation in the intestine and potentially cancer. And the other problem is skin, of course. So there are other products, IL-22 based products that are being tested by pharmaceutical companies. So this one is a dimer of IL-22 linked by an FC protein, and then there's another one which is IL-22 FC by Genentech. Both of those are in phase 2A and 1B clinical trials for different indications, including for inflammatory bowel disease and alcohol hepatitis. But in the safety work that these companies have done, all of them have reported adverse events in the skin. So you get these skin lesions, especially where the subcutaneous injection's been given. But also when given IP, you see random skin lesions appearing, which suggests that a potential higher or chronic dose of IL-22 could lead to skin proliferation, which is what we essentially want to avoid with our therapeutic. So the solution, we decided we would somehow target IL-22 to the cells that we are interested in. So initially that was just the beta cells because we were focused on type 2 diabetes, but we saw benefits in targeting IL-22 to the liver as well, especially when you take into account metabolic syndrome as a whole. So what we decided to do was join IL-22 with a ligand which would target it to either the liver or the pancreas. And the reason why we moved away from type 2 diabetes essentially is because there are a lot of current therapeutics that are already on the market. Some of these are oral orally taken. So we decided to look for other indications, which is why we concentrated on fatty liver disease and NASH. This is because there's no approved therapeutic for fatty liver disease and non-alcoholic steatohepatitis, so fibrosis in the liver. So I think Ben's going to talk a little bit about commercialization, and then we can get some questions.
Thanks, Sumaira. So as was mentioned in the introduction, I'm from UniQuest. I think everybody knows UniQuest, the commercialisation company of the University of Queensland. And so my role is, as Director of Asset Management, is really to look after the revenue generating aspects of UniQuest, whether that's from a license of intellectual property or because we have a shareholding because of a startup that we've entered into where we've licensed some IP in. So what I wanted to do was just as part of this presentation, just give you a bit of an idea of the steps, very, very broadly speaking, that were involved in commercialisation of Sumaira's IL-22 technology. So really the first step was filing a patent in respect to the technology. A lot of discussion went into this at the time, and really there's two broad aspects of things that are covered in this patent. A method of using a targeted form of IL-22 for some kind of pro-metabolic outcome, and some composition of matter claims around a whole raft of different ways of targeting IL-22 appropriately. And really the first challenge in the commercialisation of this IP was to find the right indication. So we spent a lot of time initially talking to commercial partners in diabetes. And although we could demonstrate that the IL-22 approach was non-inferior to current gold standards in terms of diabetes treatment, there wasn't a lot of interest in just yet another diabetes compound. And so it became evident that if we could generate some data demonstrating the fact that IL-22 might be useful in an indication like NASH, which had a huge market need and a rapidly expanding market, then we would be in a much better position in terms of being able to commercialise this. And so that's what Sumaira set out to do. We got a really good data package around demonstrating the fact that IL-22 was particularly useful in reducing inflammation and fat accumulation in the liver. So if I had to sort of point to some of the lessons from this commercialisation project, I'd say that one of the key things was making sure that we found the right indication and we got the right data package for that indication to get commercial interest. That was a pretty key step in being able to get the outcome that we did. And so just to say a few things about the indication of NASH, it develops from steatosis, a fat deposit in the liver, a large accumulation of fat, from which you can get non-alcoholic steatohepatitis developing concomitant with an increase in inflammation. And that can be present with or without fibrosis. If that progresses, then you get the formation of cirrhosis, where the scar tissue makes the liver very hard and unable to work. And end stage can also end up as HCC or liver cancer. So the advantages of this market for us and being able to go to commercial parties was that it was a highly prevalent disease. So it's about, the prevalence of NASH is about 65 million cases worldwide. It has enormous growth characteristics. I think the current the compound annual growth rate for NASH is something like 68% up to 2029. And what's driving that is largely things like obesity and an ageing population. Although patients with NASH are often asymptomatic and don't see any particular need sometimes to really bother too much about treating their NASH, it really is driving increasingly a demand for transplant services, which has increased pharma's interest in providing some kind of a pharmaceutical solution to this. There's no treatments currently specifically indicated for NASH. There's some off-label use of a few compounds, but not a lot of really strong clinical evidence to support their efficacy. And there's no simple test to detect NASH. So these are all really useful drivers for when you're trying to pitch something that is a treatment for something like NASH. So what we did is we developed a data pack really focused around our liver data. And we spent a lot of time pitching this around the place. And I can see around Jenny smiling in the audience here, because I remember when she did this with Dendrite, they did something like 70 pitches. We didn't have to do as much as that because this is quite a hot area. But we still did probably 18 months worth of pitching this project around the place to pharma, biotech and venture capital companies. And we eventually caught the interest of IP Group, which is somewhat unusual venture capital fund in the Australian context because it's a London Stock Exchange listed company, entered into an agreement with a group of 8 to provide venture funds to university-based projects. And so this was the first investment that IP Group made into a Queensland university for a life science project. And so out of that, we gave rise to Jetra Therapeutics, which is a purpose vehicle for the development of these IL-22 based compounds. So I thought that I'd just give you a few just general comments about startup companies and how they actually work. This is relevant to Jetra. There's the formula for establishing a university-based startup is pretty much never changing. And the way that it worked in this particular case is consistent with many other cases, in that UniQuest itself doesn't have a lot of spare cash to invest in startup companies, and most universities, particularly in the current environment, would find themselves in the same position. And so what UniQuest does is to license the intellectual property across to the new co, in this case, the newly established Jetra Therapeutics. In return for that IP license, gets a shareholding. And the value of those shares is really determined by what's known as the pre-money valuation placed on the IP. So how valuable in a commercial sense that IP is to the company and to any kind of eventual exit. And then the investors, in this case IP Group, get their shareholding by virtue of the investment that they make into the company. Now, of course, over time, because UniQuest tends not to invest into these companies, we do get diluted out, but you've got to remember that the value of the company as a whole increases over time as more and more work is performed. And so we eventually, at exit, will end up with a smaller proportion of something much more valuable than what we started with. So just a few words too about the company structure. In companies like Jetra, we use a very simple structure to try and keep the costs down as much as possible, because there are some costs just in running a company, and really just need to minimise those as much as we possibly can. So initially speaking, we only have two shareholders in Jetra. We've got the IP shareholder, which is UniQuest and IP Group as the first of the investor shareholders. We have a small board, and then we have contractors and consultants that do the work. And so MARTA benefits from this because under this arrangement, all of the research work that's been done to date as a result of the startup has been contracted back to MARTA. There's only a little bit of synthesis work which we've done at GenScript. And then we use some consultants as well, and particularly now as we're starting to look at the next round of investment. But as the company matures, we'll look at adding in a management team, particularly in the next round of investment, some project managers that can give some guidance to the work that's happening. And then only very lastly, when this is a more mature company, will we employ staff. It's really only when you have a significant amount of investment into the company that you can start looking at employing people. And so in terms of how anyone ever extracts any value out of this, it really needs to be at exit. And so UniQuest will eventually realise the value in this arrangement at the point at which the company is sold. And commonly, and generally speaking, this happens through a trade sale. And, you know, in terms of Jetra, the sorts of companies that would be interested in acquiring the asset would be ones that are already playing in that NASH area. And there's quite a number of companies. It is a busy pipeline. But also we have done initial public offerings in the past when the company is listed on the stock exchange. Most of the UniQuest companies have done an ASX listing, but we have done NASDAQ as well previously. But there's a whole host of different options when you look at IPO. Probably not a favoured outcome of venture capital, but still a possibility. So I just wanted to hand back now to Sumaira to let her just sum up by talking about some of the things that she's learnt from working together with UniQuest and Jetra.
Yeah, so I think the main thing that I did learn is that we need to find the right indication, like Ben said. So we had followed, after filing the patent in the PCT in 2015, we followed the route to look at a type 2 diabetes anti-diabetic sort of drug for about 2 to 3 years. And so this just means that the life of the patent is slowly decreasing as well. So I think if we had found the right indication at the right time, we would have a lot more time as well, a lot more data, a better data pack by now, now too. So I think finding the right indication is really key. I guess I don't want to say numbers don't really matter, but type 2 diabetes, very prevalent, but you need to look at what the pipeline has at that time, because if there are really good drugs, especially if there's orally available, you know, you take it once every 2 weeks or something, sort of drug, then it's unlikely that the clinicians are going to incorporate a brand new unknown biologic into the clinic. So I think it's important to find an indication where there is a big gap in the market as well. Also, another thing to note is time commitment. So doing this does take up a lot of time, especially when you're doing 20 plus pitches in 18 months, as well as grant writing, etc. But it does mean that each time you do a pitch, you do improve your data pack to focus it on a specific thing that's probably lacking. And all this stuff that I consider behind the scenes, which Ben does obviously, so talking to the patent attorneys and filing divisional patents and things also takes time and commitment, obviously from a UniQuest point of view. And moving forward with the therapeutic development plan, you know, that takes a lot of time to understand and build on as well, what IP group is looking for, what Jetra is looking for, and what the clinical partners are really saying that you need to incorporate it in. So that takes a lot of time. Also, we need to have a think about gold standards. So the problem with NASH is because there aren't any therapeutics, anti-NASH therapeutics in the market, finding a gold standard or a competitor is really difficult, and a lot of the different companies don't agree on what the competitor should be. So this adds to the complexity. But it does really help with attracting more funding, especially for my academic research as well. So we've been able to secure an NHMRC development grant previously, and now we're in the process of putting another application in. Also helped me get an NHMRC ideas grant. So when I guess reviewers see that there is translational impact potentially, that does add to benefits in terms of attracting funding. And also it's a really good extra stream of money coming into the lab. So Jetra is subcontracting most of its work to MARTA Research in my lab. So this provides us with a good stream of funding coming in, and we can see the larger picture as well in terms of what we want to achieve in the end. So that's all we have today, and then we were just going to answer questions if there were any. Thank you.
Great, thanks. That was fantastic. And I had the privilege of being very briefly involved in one of those pitches. It was right about the time where they were getting strong feedback to switch indications, that the diabetes pathway was not the right pathway to be going down. It was, that was not easy for Sumaira to give up that passion, which has been her research focus. So it is a really good lesson to have learnt, and something they might want to ask us some more questions about. So who does have some questions? Yep.
That's such an interesting journey. How did you come up with the name Jetra?
Oh,
It's Slavic, and one of the investment analysts.
Yeah, come up, Ben. This is,
Yeah,
we've got online people, so.
Yeah, so yeah, it's Slavic for liver, and one of the investment analysts with IP Group, that was her ethnicity. So we went with that because we couldn't think of anything better.
we came up with lots, but they all included IL-22 or liver, and it just didn't sound as good. So I think Jetra was a good choice.
Norbert?
Great talk, and the journey, anybody talks about a breakthrough, this is a long haul getting something done. Now, the success of a company can be sometimes be judged by how many patent attorneys they employ to actually defend the sharks, then, claiming, or, this is not you, or whatever, this is our. So when you do the initial searches and big operator prior analysis, I guess it's how easy or hard is it to ensure that if you switch into a new indication, that there's no prior art, or you have freedom of operate.
Do you want to just paraphrase that really quickly for the online recording, just in case?
Yep. So one of the tricks to filing IP can be searching the prior art, or all of that body of literature that's already present that might disclose your invention. So how do you make sure that you don't miss something is really the question. Look, I suppose that's where the skill and expertise of UniQuest comes in. We've been doing this for decades now, but we also have on staff a couple of attorneys that help with that. All of the drafting work is done by external counsel, but yes, it is always possible to miss something, particularly because the test is something published anywhere in any language. But, I suppose it's easier when it is an idea that hasn't been extensively published on anyway previously. And I suppose one of the ways that we ensured that there was a lesser risk to us was to take on this targeting approach, because an IL-22 based therapeutic that wasn't targeted probably would have been more difficult to navigate around the
You
arc.
want to make sure you're in Iceland. Iceland is on your countries of. Is there the story that Iceland was missed once with heparin race?
Ah, yes.
And the stars of Iceland.
Yeah, well, I mean, later on down the track, choosing which countries you're actually going to file in is another trick too, and there's a whole lot of different considerations that come into play there. Yeah.
I would have called the company national too. But no, a serious question is, amongst your 20 features, you mentioned that NASH has no good test available.
Yes.
So we'll rather speak against it rather than.
Well, it's not no good test available, no easy test available. So
Still, you have a therapy, you're proposing a therapy, but you don't know what for because you can't test it. So I'm just being devil's advocate. It obviously works for you guys, but
that's true, but it also creates an opportunity, because if you can find a biomarker as part of this work, then there's an enormous market for that. So it's not that. I mean, the diagnostic test is FibroScan, which is an ultrasound based test, plus a liver biopsy. So they're complicated procedural tests rather than a simple blood test. That's more the point.
So I guess the people with NASH, they get picked up a little bit later on in the stage, and that's why we're trying to target the fibrosis in the liver, because by the time people actually present to the clinic, they do have fibrosis in the liver. So that's what we really need to achieve is for IL-22 to work there.
Sorry,
Just a Q&A, and I think it's trying to answer the question
yep.
as well.
can we? Where's the mouse gone? Can you? Oh, there it
Yeah,
is.
there you go.
Yeah.
Oh, yeah.
Oh,
Stop sharing,
no.
stop sharing.
It is a question.
There we go. So any more questions? Right, okay. Ranjini started already, Dan?
You mentioned all the money, which is great. And I wondered how you're managing that. Do you have a project manager? Do you have a CEO? How are you kind of, because it does take a lot of time for you, Sumaira, to get your head around so many things and to manage so many people and doing experiments for you to do at a time with patients. How do you manage?
So I guess we were only in the pre-stage seed, and we didn't really know if we were going to move forward with the seed stage. So at that time, I was doing most of the management along with Ben and IP Group. That's on the board of directors for Jetra. It did take up a lot of time, and I think it was a learning curve for me, a very steep one as well, because I had to do all the budget management, which is very tricky actually, especially when there's 3 separate entities as an IP group and Jetra, of course, but then UQ and MARTA. So it makes it quite tricky. So it is a learning process, but I think for the next development plan, we have all these things incorporated in, and Jetra is thinking of bringing a sort of project manager on board, which will make it quite a lot easier and more streamlined as well.
I think I went the chat, not the Q&A, but still the same one. Maybe it is the chat. Nope, same question. All right, we're all good.
Oh, no, it was just,
Did you know?
did you know from the outset that this was really a goer?
No.
No.
That's pretty definitive answer right there. I think we need to expand on that. Yes.
Can I just explain a little more about the scope of your IP and what is protected?
Okay, yep. So there's 2 broad sets of claims in this patent, which one is, some methods claims around a method of targeting IL-22 to the pancreas using some kind of targeting moiety. So the work that Sumaira has presented today, and the lead in Jetra, is a SCFV approach to doing that, where the fragment targets a cell surface structure on the pancreas. But that patent also talks about, using GLP, for example, as a way of targeting. And the second set of claims in that patent is to do with compositions of matter. So specific examples and sequences associated with protein fusions that target IL-22. And there's a whole raft of them in there. And that's pretty much a common structure for patents like this.
We're really short on time because we've already gone over, and I just want to give Scott an opportunity to ask.
I have two questions. One was, can you give us a sense of the support you have to research and taking a slightly different approach? And secondly, when your CDF runs out, I think this is a question more for Angela, how do you sell the story about this dual direction you're taking to get a leadership fellowship?
Yes. Oh,
just written one
that's
of those.
very, yes. It's tricky because it does prevent you from publishing things in time for applying for investigator grants, I think. So I think it has delayed some parts of the research. My strategy has been to be really broad in the research I do. So I work on inflammatory bowel disease, viral infections in the lung, CF, asthma, etc. So I think that's been my strategy, is that keep doing that sort of work as well, so I can get my output from that sort of stream. So eventually it doesn't impact it that much, and I can make a case for the commercialization and translation. In terms of the support, I would say that Marta's been really supportive, and Marta's here. They've been really supportive, and I've not encountered any issues in terms of me taking this forward as Jetra Therapeutics and getting subcontracted research done in the lab.
Well, it's part of our vision and mission as an organisation, so it's completely aligned with what we want to do. So of course we've been supporting it, but it should be acknowledged that Sumaira just works incredibly hard. She's extremely committed to it, and at the same time juggling small kids at home with a partner who works way a lot, etc. So she's really got to be given a huge amount of credit for the success of this, along with the UniQuest support, who really stuck with her through the point where they were getting so many no answers on the diabetes indication. So I think it's it's a good story of resilience. Any more really quick final questions, because we are now 10 minutes over. No, excellent. Thank you so much. Oh, no, wait.